Here we go.
Okay. Okay, guys. Wait. Thank you. When do I put up the slide? I put them up right before lecture every day. Yep. But I would love it if you guys did not use tablets, laptops, all those things. If you must, I ask to stay in the back or somewhere where you won't distract or on the side. I understand everyone's different, but if you can avoid having something distracting with different alerts in front of you, that'd be great. Okay. Quick business. Pre-test. The average was 83, so that's fantastic. That means most of you guys are ready to go on the ball. Everyone but 2 people took it. The 2 people who didn't take it probably just forgot. So, those of you guys who are forgetting things, there's a lot of people forgetting quizzes already. Please get that under control now. It will bite you in the butt later. Okay? So, like right now, should you be, you're like, oh my god, I never put my reminder in my phone. What did I put? Or if you like ignore your reminders, or like your alert sound is something that you need to change to make it a little more shrill, maybe do that. I don't know. Let's not let this be the reason you get a C or lower. Okay, so keep an eye on your average. The other thing is, in my grade book, it's got like running. Everything I do is on Blackboard, so everything I see is what you see. So if something's not right, email me. Other than like, oh, I don't have an A. I mean, yes, yeah, that's not right, but I can't fix that. But keep an eye on your average. There are already some people who have like 40% averages because they're missing quizzes. And so you want to get that under control now, not the last week of semester. I'm just going to bother people a lot this time around because it makes me want to cry when someone who should be rocking it with an A doesn't get that A. Okay, don't forget to bring Scantron's presentation. Yes?
Do you post the presentation for the grades? Or?
It should be posted.
Oh, it is.
Yeah. If it's not posted, email me. There might be something on your Blackboard, or I don't even know. But everything's posted. I posted it about 11 o'clock last night.
Yeah, I can check.
Yeah. Okay, but everything, I post every single thing that you do in this class, so nothing should ever be a surprise. Okay, so let's take 5 minutes to write. Okay, so oligosaccharides are added to proteins in the ER. This is where the glycosylation found on cell surface receptors, originates. What role do these sugars play in protein folding and quality control? So take a minute. You can always look at your notes to kind of refresh what's going on. Okay. Do you guys feel like 5 minutes is good or too long? Should we go down to 3 minutes? Or are we feeling good about 5 minutes? Okay, seeing nodding heads. Okay, so let's see. Who's going to tell us what they found? How about Elizabeth Lane? Liz Lane? There you go.
Okay, to be honest, I don't really know, but I kind of took a guess. I knew that polysaccharides are like polymer or simple sugars, and I just kind of said that they play a role by ensuring the protein grows properly, but I don't really know.
Okay. So on an exam, that would be wrong, but that would be like your introductory sentence.
Okay.
And then you would go on to fill it in with
Right.
now. So, and I like getting these kind of answers too because so many times on exams I'll get that and that's it, and that's never enough. But you know what? You're brave for just coming out and saying it, so good for you. So I'm gonna just check you off so you know that I won't ever have to call you. Yeah.
Okay, I wasn't positive on how to answer it, but looking through some of my notes, I thought I might have pulled some keywords that have to do with like the ERAD and UPR and the calnexin and determining like the following cycle on like which ones are allowed to leave the ER and which ones have to be sent back.
Right, so you're on the right track. One mistake that you made was grouping UPR and ERAD together.
Okay.
So stick with the step before ERAD in that folding cycle
Okay.
and calnexin, and you're on the right track. Who wants to share more? Yep.
Says that with protein folding because the protein folding cycle consists of a series of binding and cleaving sugars, and that once the protein is properly folded, it will no longer bind calnexin, and calnexin and calreticulin are what holds it in the ER. So when it can't bond to calnexin, it's released.
That's perfect. That's a full credit answer. Right, so the sugars, the whole folding cycle has to do with the trimming and adding of sugars and whether or not you have one sugar conformation or over another determines whether calnexin will bind to the protein and retain it in the ER. Right, calnexin binds proteins, keeps them in the ER until they can fold properly, calnexin and calreticulin. And as soon as the sugars that dictate whether or not calnexin can bind are cleaved, then that protein is released either to go to a vesicle and go to the Golgi, or is retrotranslocated out of the ER, which is ERAD. Okay. Yes.
So what's the exact difference between calnexin and calreticulin? They both do the
I
same
believe
thing?
so. Calnexin is the difference between calnexin and reticulin. That was a question. Calnexin is a membrane protein that is anchored in the ER membrane. Calreticulin, I believe, is not, but they both bind sugars and both participate in folding. And whether or not they can bind the protein depends on the sugars that are on the protein. Okay, good question. Okay, any other questions? We're going to start today with kind of going over ERAD and into UPR in more detail. Okay, good. Yes.
Could you explain the role of cytosol exactly?
So, are you questioning exactly what cytosol is, or
Well, because I was confused on the quiz because I thought that right after the protein enters the ER goes to your cytosol and then to the protein.
okay? So the question is, and this is you're not the only person who asked me this question. So the question is, what do I mean when I say the protein is in cytosol, right? And so what I mean by that is the protein physically itself is sitting inside the cytoplasm. The cytosol, same thing, right? Normally, so the secretory pathway, as we'll learn today, once you enter the ER, you never are in cytosol ever again. You are going to bud out of the ER in a vesicle, so you're surrounded by membrane, and then you're going to transit through the cytosol, so the vesicle itself, so it's like in a little bubble, that's going to transit through the cytosol to the Golgi, and then from the Golgi to wherever it needs to go. The protein itself never leaves the ER unless it's through ERAD to be degraded. Yep, and that's why I believe in that question I said properly folded proteins, so that you didn't go down the ERAD route. Yep. Any other questions about that? That seemed to come up a few times. I got at least 2 or 3 emails about it. Okay, so far what we have done is we're talking about intracellular traffic. We started with the secretory pathway. We talked about import of proteins into the ER and all the different parts involved in that mechanism. We spoke about integral membrane proteins and how they get into the membrane at the ER, and about the transmembrane domains and stop transfer sequence and all this good stuff. We spoke about the processing and modifications of proteins in the ER, such as disulfide bonds and stuff like that. We've been talking about protein folding and quality control. That was what our little writing assignment was about today. And we ended talking about the ubiquitin proteasome system. And so we're going to kind of pick up with ERAD and the ubiquitin proteasome system. Right, so this is what we were talking about in our writing assignment, right? So the state of this end-linked glycosylation senses correct or incorrect protein folding in the ER. And if it's incorrect, you get retrotranslocated out of that translocon, and that's called ERAD, ER-associated degradation. And so these are these sugars that are added on, and depending on the conformation, notice you have 3 glucoses. Here you have 1 glucose, here you have none, and it just so happens calnexin wants to bind to the 1 glucose version of the sugar. That's what it can bind to. When it has the 3, it won't bind, and when it has none, it won't bind. And so that's retaining it in the air so it can fold. And so that cleaving and adding of sugars is that folding cycle. A normally folded protein is going to exit the ER in a vesicle. A protein that can't be folded will be retrotranslocated and degraded. So this is just another figure from a paper showing ERAD. So again, if you have prolonged association with these ER chaperones, such as calnexin, calreticulin, then you're going to be removed from the ER and degraded, because that suggests you can't fold properly. And so this is just kind of showing the same thing, a different way that you've seen it, is here's your RNA and your ribosome, and here you're being translated into the ER, and then those sugars are added, right? And so then what you're going to have is binding to calnexin, and there's going to be folding going on. And if you fold correctly, here's your vesicle. You go out in the vesicle towards the Golgi. Now this, you could easily think from this picture that you might pass through the membrane to the Golgi, but that's not what happens. That arrow is drawn in a confusing location. You are actually inside the vesicle, because a protein can't pass through a membrane, right? But if you're not folded properly, you'll go through this cycle again and again. And if you can't fold ever, then you get exported out through a translocon, and then you are ubiquitinated and degraded via the proteasome. And so this goes into the detail of exactly what sugars are added and what is trimmed. You don't have to memorize that. I don't like it when you have to memorize things, because then you get so stuck on the details, you lose sight of the big picture. But sometimes detail helps to understand something fully. So these are these sugars that are added onto the protein in the ER. And so they're all different sugars, and so you can kind of see the different sugars that are removed, they mean different things. And so you could think of this as the glycan code, right? If it looks one way, it means one thing. If it looks another way, it means another thing. So you'll get some glucoses that will be removed by a glucosidase. You can guess from the name of that, glucosidase. It's something that's going to cleave glucose, right? And then you've got this monoglucosidated, so one glucose protein that is recognized by calnexin and calreticulin. And that's what's retaining the protein in the ER. And that facilitates folding. And so then when you have removal of this final glucose, because this glucose here, that's a L, the L-glucose, that's the one that's required for this calnexin calreticulin binding. And as soon as that's removed, it can't bind anymore. And so it'll either exit if it's properly folded, or it'll be added again. So if you'll be re-glucosylated, and then rebind calnexin calreticulin and re-enter that folding cycle. And then if it's not meant to be, then removal of this mannose here will signal for exit via ERAD. Okay? Okay, so misfolded proteins are translocated into the cytosol, so they go right through that translocon. They're ubiquitinated, they're degraded via the proteasome. So you might say, who cares? So I mentioned the other day there are some diseases associated with the ERAD. So here's 3 examples. One, cystic fibrosis, which I mentioned the other day, where CFTR, I couldn't remember, CFTR, cystic fibrosis transmembrane conductance regulator, that there's one amino acid change, and it causes a failure to fold, and it's targeted to the proteasome via ERAD. And then in juvenile Parkinson's disease, there's inefficient folding of a GPCR, a G protein coupled receptor. We'll learn about those receptors before you know it. And that leads to ER retention, right? If you're not folded properly, you're retained in the ER. Then they get aggregation, and in this case, actually, the unfolded protein response is triggered, and then you get apoptosis. Because Parkinson's cell death, right? And then CMV, cytomegalovirus, it promotes degradation of MHC class 1. Does anyone know what MHC class 1 is? I'm hearing antigen. It has something to do with the immune response. It has something to do with the antiviral response and recognition of virally infected cells by the immune system. And this virus promotes degradation of that protein. So that's kind of awesome, right? Who thinks that's awesome? It's not, thank you. It's not awesome for us, it's awesome for the virus, but it's kind of cool that the virus is like, ooh, you are going to be what gets us recognized by the immune system. Let's get rid of you. A lot of viruses do that. And it does that through retaining MHC class 1 in the ER, and then it gets degraded by ERAD. Okay, so the other day someone said, what is ubiquitin? What is the proteasome? So I've got a small intro about that, and for those of you who don't know me, I love the proteasome and everything proteasome and ubiquitin-like. So it's definitely best to think like that, because my questions on exams might be geared to those kinds of things. And it's super important, right? Right. So this is this protein tag. That's a small protein that's covalently added to other proteins. It's a post-translational modification. People always get confused. They always don't know what a post-translational modification is. What does that mean? Something. Someone raise your hand. Or they'll call on you. Yes.
The modification is added after it's translated?
Right, a modification added after it's translated, and it's usually covalently added, meaning it's not just something binding, it's something that is permanently attached. So if I add things that denature proteins, it will not fall apart, it will not fall off. It's present in all eukaryotes. It's conjugated to lysines in the protein, and there are many functions, many functions, only one of which is targeting proteins for degradation via the proteasome. So these are the different functions that I mentioned the other day. So there can be different linkages of ubiquitin chains because they're polyubiquitin chains. So ubiquitin bound to ubiquitin bound to ubiquitin, and the way they're linked could mean different things. So you know how we just spoke about the glycan code and how the shape of the sugars means something, and whether or not calnexin and calreticulin combine? Well, it's kind of the same with ubiquitin. The shape of the chains determines what can bind to them and what can happen. And so it means a lot of different things. And so what we're talking about with ERAD is this lysine 48 linked polyubiquitin chain, which allows that protein to be recognized by the proteasome. But then there's also other kinds of ubiquitin modifications, and I'll try to point them out as we come across them in this class. But a big one that has to do with what we're doing now is you can get monoubiquitin added, and that will signal endocytosis. That has nothing to do with the proteasome, right? It has nothing to do, well, it does kind of have something to do with degradation, but more via the lysosome, and we'll talk about that soon. Also, some kinds of ubiquitin chains are going to activate signaling pathways. It has nothing to do with protein degradation. I'd just like to hammer this in because a lot of people come out of biology thinking protein, misfolded, degraded. That's what ubiquitin does. Way bigger than that. So this is the ubiquitin cycle. So it's an enzymatic reaction that requires 3 different enzymes, conveniently called E1, E2, and E3. And so one just activates ubiquitin, then it's transferred to E2, which then binds to or transfers ubiquitin to E3. E3 is the most important one, though, because the E3 is what's going to bind to the substrate. That's the protein that needs to be modified or degraded or whatever. So if something's wrong with your E3, you might get a lot of proteins that don't get degraded. That could be really bad depending upon the protein. And you see this in cancer and stuff like that, where cyclins that should be degraded are not being degraded, and then you'll keep progressing your cell cycle, for example. So then the polyubiquitin chain is recognized by the proteasome, which is this barrel-type protein structure. There are tons of, there's not one proteasome, there's tons of proteasomes in the cell. And then the protein's going to be degraded, and then those ubiquitins will be recycled to be used again. So if there's one thing you leave this class with, it's that ubiquitin does much more than target misfolded proteins for degradation via ERAD or just in general. Do not limit yourself to thinking ubiquitin, ERAD, ubiquitin, misfolded. No, way bigger than that. And so this is just something I pulled off some website that shows just the different cellular processes that are regulated by ubiquitin. So you've got everything from DNA repair, cell cycle, transcriptional regulation, the immune response, signal transduction, metabolism. It's all regulated by ubiquitin. And then if you start adding ubiquitin-like molecules into it, there's even a greater level of regulation going on. So if I haven't convinced you, I don't know what I just did. Okay. That's enough. That's my rant and rave of the semester about ubiquitin. Thank you for coming. Okay, so the unfolded protein response. And so one of our answers today kind of grouped unfolded protein response with ERAD. And I'm so glad you did that because it allows me to say, hey, these are very different things. Yes, they're both involved with misfolded proteins, but that's where it ends. So ERAD is about that one protein that couldn't be folded, being retrotranslocated, ubiquitinated, and degraded. The unfolded protein response is about an excess of unfolded proteins in the ER. And what happens is, well, there's a few different pathways within that unfolded protein response, but the one that I'm going to focus on is the one where a transmembrane kinase that's in the ER, it senses the unfolded proteins. And then what it does is it cleaves, it basically activates an endoribonuclease. What's an endoribonuclease? Pick it apart. No. You're right about nuclease. Yep. Takes off part of the ER. Nope. So endoribonuclease. So with ribo, so remember ribonucleoprotein we were talking about in recitation last week? Who was raising it? RNA, right. Endoribonuclease. So it cleaves out some part of RNA, in this case an intron. Right, so it activates an endoribonuclease. So that's what's happening here, right? So here's your unfolded proteins in the ER. and they're being sensed. And then there's this kinase domain as part of these sensors. And so they're going to activate an endoribonuclease, which will splice out this intron here. And so then you get your spliced RNA, which comes out, makes a protein. That protein is a transcription factor that goes back in and activates expression of these genes that are involved in protein folding, such as chaperones and stuff like that. And I'll go through this. There's another figure that will go through this once more. Question?
I have two questions. So if I have the ERAD that's already degrading unfolded protein, how could you have accumulation of unfolded protein?
So the question is, if ERAD is taking care of unfolded protein by inducing degradation, then how would you even have an accumulation of misfolded proteins, right? Well, so there's multiple things that can be going on here. First of all, you never want to have one safety net in the cell, right? If you have one safety net, if something goes wrong with that safety net, then you're a goner. So that's the first thing. Second of all, you have to ask yourself, well, what's causing a protein to misfold, or what's causing a massive amount of misfolded protein? So you can have a mutation in a gene, such as this CFTR in cystic fibrosis, that's going to cause that specific protein to misfold and exit via ERAD. But you can also have some more global effects, such as some kind of drugs. The best one I know are cancer drugs that will cause ER stress. And then what happens is everything goes wrong in the ER, and you have this huge number of misfolded proteins. So it's not just there's a mutation, this protein doesn't fold, it's all the proteins in the ER are not folding. And ERAD cannot get to it fast enough, and so there needs to be an additional response. We need to increase the number of genes that are going to help to deal with this, and that's the unfolded protein response.
So the UPR is that transmembrane kinase.
UPR is the whole system, but the transmembrane kinase is one part of it. Yep. Questions? Yes. Can ERAD and UPR happen at the same time? Yes, absolutely. Yeah. Yep. So you have to think of science, and not science. Well, science is big. You have to think of cell biology in the sense of not one thing happens at a time. Often everything is happening at the same time. Yep. Okay, so we'll go into this a little bit more, right? So there are different proteins that are in the ER membrane, and their job is to sense accumulation of misfolded proteins, and what they do is signal to the nucleus to activate transcription of genes involved in protein folding. That is, like, if you had to define UPR, that would be your definition. But the real question is, don't just define it, understand it, right? So here, there's 3 different sensors that the textbook talks about, and if you kind of are sitting here going, oh, I don't know what she's talking about, it's figure 1255 right in our textbook, and you can go look at the figure and you can read the accompanying text, and usually sitting there and reading and thinking will kind of get you to where you need to be. I guess that's just life in general, right? But basically, so your sensors are going to be on the lumen side of the ER, and then your part that's going to activate something is going to be on the outside of the ER, right? And so you're going to get phosphorylation and activation. So the one I just talked about, you get phosphorylation and activation of this endoribonuclease, right? And then you're going to get the translation of that protein, which is a transcription factor, which results in activation of gene expression of proteins associated with protein folding, right? And then there are these 2 other mechanisms. But let's just focus on that one I just talked about. And so this is right out of the textbook, and it's a really nice figure that walks you right through it, step by step, right? So you've got your misfolded proteins, and then you've got your sensor here. And so once your misfolded proteins are sensed, you're going to get activation and phosphorylation of your sensor, and that activates this endoribonuclease. And so then these pre-mRNAs are sitting around because they're there so that if they're needed, boom, splice, ready to go. And so what's going to happen is it's going to splice out that intron. And then you have an mRNA that can exit the nucleus like all other mRNAs, right? Do mRNAs always exit the nucleus? Sure. Why not? And then get translated to make a protein. And so that protein So that protein is then going to enter the nucleus, bind to some promoter, and this is where you're going to have to stop me if I'm talking too molecular for you. But remember, promoters activate gene expression. Does everyone remember that from genetics? Right? And so you're making a gene regulatory protein that's going to bind to a promoter, activate transcription of genes associated with protein folding, such as chaperones. That is the unfolded protein response. Okay? It can be as complicated or as simple as you want. Questions about this? Just conceptually? Yes?
So are they, the basic purpose is just to make a protein that's integrated?
No. So the basic, no. Okay, so you're connecting ERAD and UPR, right? I think you are. So the basic point of UPR is to increase expression of chaperones. And so, but there needs to be a way. So what you're dealing with, you've got a sensor, and then you've got making of a transcription factor, which then goes and activates expression.
And that expression is for the synthesis of chaperones.
Expression of chaperones, exactly.
And why do we want to make more chaperones? I mean, like, is it chaperones fault that the protein is folded, or could it be a mutation in the protein?
So I'm saying, oh, this activates expression of more chaperones, and what I'm hearing here from, oh, I got it. I was like, nah. No, like Netta, from Netta. I looked at everyone's pictures yesterday. It was very fun. Posted them on Facebook. Yeah, I made smiley face men. You guys see that movie One Hour Photo? I promise it's not like that. That's all I'm saying. It was one of Robin Williams' best movies, and no one mentioned it when he died. And I was so upset because it's like that was like, because it showed his range, because he played kind of a psychopath. He never plays a psychopath. I mean, Mork and Mindy, that was kind of crazy. Anyways, okay, I digress. Sorry, Netta. So the question was, are you just trying to make more of the protein that got, oh no, the question was, why are you trying to make more chaperones, right? And so with ER stress and this massive accumulation of misfolded proteins, it's most likely due to some external issue, not just a mutation, because remember, you're not translating one protein at a time. In the ER, there might be 50 kinds of proteins being translated. And so it's more of a global effect that's going to cause to activate the unfolded protein response. And so having more chaperones will help to fold these proteins. Okay, yep.
Did you say the pre-mRNA is just floating around in the cytosol?
Yeah, so actually the pre-mRNA is already there, not in the cytosol. It is, so here I'm actually not sure that I'm thrilled with how they're showing it, because remember your ER is contiguous with your nuclear envelope. I'm having a hard time with that. And then it's going from mRNA to protein without any sort of, so it's possible that it is floating around the cytosol. It's floating, think of it this way. It's already there, and it's in close proximity to the ER. So it's either in the cytosol or in some area that's near the nucleus and cytosol. Because you would have to ask, well, how did it get out of the nucleus with an intron? right? Because you shouldn't be able to leave the nucleus with an intron. But there are actually ways to get out of the nucleus with an intron that a lot of viruses use. Like sometimes there could be a protein that might cover up and export the RNA out of the nucleus. But yeah, I had the same problem with this figure, so good catch. But really the main point is that it's there, and it's spliced, and then you make that transcription factor, which then can activate gene expression and get more chaperones made. Okay, I'm going to move on unless anyone has any pressing issues. Pressing issues? Okay.
Okay, so I'm confused as to how the transmembrane kinase became the ribonuclease. So is it just the phosphorylation that turns it into that?
Yep, so transmembrane kinase, so okay, here's the deal. It's a protein that has different domains, so different parts with different functions. It has a sensor function, it has a kinase domain, and it has a ribonuclease domain, right? The kinase domain will not be active until they come together. Or the kinase domain, yeah, and once the kinase domain, no, it phosphorylates itself and it must come together, and then when the ribonuclease domains come together, then they're active. So when it's apart, it can't be active, but when it's together, it's active and able to splice out the intron, and that's determined by the phosphorylation.
So you need two kinases to have an active ribonuclease domain.
Yes, you need two kinases. There's two sensors come together and activate each other, and that makes an active ribonuclease domain. And that can cleave the intron.
And that pre-mRNA that's in the cytosol is specifically destined for that pathway.
The pre-mRNA in the cytosol is the RNA for this transcription factor, this gene regulatory protein. It is a very specific, yeah, it's not all okay, thank you. It's not all RNAs. It's a specific RNA that encodes this gene regulatory protein. Yeah. Okay, moving on. Okay, here's our summary slide. Protein folding in the ER, right? So proteins come in, chaperones bind, disulfides happen, they're rotated around, they're peptide bonds, glycosylated, folded. If things go wrong, ERAD. I guess I should have put if things go really wrong, unfolded protein response. So it's always good. So right now I just gave you all this stuff. It's always good to be able to fit it into one picture or one story. It's all related. So if you're having trouble relating these items, you need to be seeing me now. Well, not right now because I'm teaching. Yeah. In a normal cell, so the question was, in a normal cell, would ERAD be the way to get rid of proteins, and only in extreme circumstances would the unfolded protein response be activated? I think of it as quantity. If there's a large quantity of unfolded proteins, then unfolded protein response gets activated. And if there's a small quantity, and it's a protein that could never fold properly, then ERAD handles it. But then sometimes there might be a lot of some protein that ERAD is trying to handle that will activate the unfolded protein response. So don't think of it as normal cell, abnormal cell, just think of it as depends on quantity of unfolded protein in that ER. Yes?
So UPR is more trying to get all the proteins to fold than degrading them.
UPR is trying to encourage protein folding rather, yeah. UPR is not degrading any proteins. Yeah. Yeah, UPR is about activating gene expression. Okay. Yes?
Something about UPR apoptosis with like cystic fibrosis.
Right. No, that was Parkinson's, yes. So unfolded protein response can't, so many genes are activated, not just chaperones, that's my example, but you can get, if you cannot fold, you have this continued ER stress, then apoptosis, cell death, yeah. Okay. Good. Okay, let's move on to the Golgi, right? So properly folded proteins travel from the ER to the Golgi for further maturation. I have to say, I love all these questions and all this, because for me, it's less important to get through massive amounts of material than for everyone to really get what we learned. So if all we ever learned is ER to Golgi all semester, I would be fine with it. I'm sure the department would not, but you guys would come out solid. You would get it in such a deep way versus like trying to cram all this stuff down your throats. And then, so I love it. Always ask questions. Okay. So ER to Golgi, remember this is part of this whole pathway, the secretory pathway. All right, so proteins are going to move between compartments in the secretory pathway by means of vesicular transport. So notice here that, so this could be your ER, and let's say this is your Golgi in this example. They're calling it donor compartment and target compartment because they're different compartments. You could be going Golgi to lysosome, right? But in every case, you're budding in a vesicle, and then you're traveling in a vesicle, and then you're budding with your destination compartment. And so that's why the answer in that on the quiz for cytosol is wrong, because notice no proteins are ever in the cytosol, right? They're always in a vesicle. Okay, so we've been talking, yeah.
Would that vesicle be considered the ER? Or the ones that run from the Golgi at traveling would be considered the Golgi? What would they be?
So you're wondering, how do you determine what compartment is what? So, did you read the paper for recitation yet? Okay, so we can kind of touch on that a little bit in the paper for recitation. So really, it has to do with what proteins are there. So the ER is going to contain all the proteins that work in the ER, and the Golgi is going to contain all the proteins that work in the Golgi. And vesicles are going to be sent back with proteins that belong in the ER, and proteins, proteins that should be in the lysosome are always going to go there. And the vesicles are transiting in between, and so sometimes you can identify the origin of the vesicle based on the proteins that are on it. And that's kind of, so you'll see we're going to talk about RABs. There are different RABs coming out of different compartments, and that's involved in vesicle fusion. Snares also are specific depending on what compartment you're coming out of. So if you were a scientist and your job was to find out, well, where is this vesicle coming from that these proteins are in, you might start to look for snares and RABs and specific things that you know should be there. And that's what they do in the paper that we're reading in recitation this week. Good introduction of the paper. I'm going to have to plant those kind of questions more often. Okay. Questions? Yes?
Is compartment just like another word for organelle?
Compartment, you can think of as location in the cell, organelle in this case. Yeah. Yeah, people often will talk about when they're studying where proteins are going, they'll refer to it as what compartment the protein is in. Yeah.
So, these vesicles have receptors that would send them to these particular target compartments?
The question is, are there receptors that send vesicles to where they need to go? Yeah, so they're not receptors, but they're proteins with similar functions that are involved in targeting vesicles to where they need to go. And we're going to talk about that. Those are the RABs and the snares, and that's all very specific because there's certain pairing. The snares need to pair in a certain way in order for fusion to happen, and then the RABs are going to target for the vesicles also. Okay. Good. Great questions today. Okay. Great. So we've been talking about the secretory pathway, but here I'm going to introduce the endocytic pathway because it's kind of the reverse, right? So if you're headed outwards, you're the secretory pathway, secrete, right? Out. Endocytic, endocytosis, come in, right? So you can think of this as just one huge system, right? Of the flowing of proteins out of the cell and the bringing of proteins into the cell. It's all using that same, the same machinery in a way, and a lot of the same concepts of moving around in vesicles. And so I'm introducing it now because we're going to be talking about proteins going to the lysosome, and we'll be talking about it in two ways. And I always lose people here, so I'm pointing this out. We'll be talking about it in terms of the secretory pathway, where proteins that are being made, so it's more of a biosynthetic pathway, proteins that are made that work in the lysosome are going to go through the secretory pathway to the lysosome. But then there's proteins that are coming into the cell through endocytosis, say from binding to a receptor, and then you have endocytosis and you form the endosome, and that goes to the lysosome. So there's kind of two ways that proteins can get to the lysosome, and I want you to kind of keep in mind that those two ways exist, and when you're thinking about it, try not to confuse them. Okay, so transport vesicles, they have different codes with different functions, but there's one basic theme. So people always ask, do I need to memorize which coat goes where? No. Just understand the purpose of a protein coat in vesicle formation. Basically, without that coat, you wouldn't have vesicle formation. But, going back to your question, just the fact that different vesicles are involved with transport between different places, you can start to, coats are involved with transport between different places, you can start to look for the different coats and know where your vesicle is coming from. So if you were working in the lab and trying to find out where your protein of interest was going or coming from, you can do immunofluorescence and buy antibodies that detect clathrin, COP1, COP2, and stain and see where's your protein and what coat is there. And then you can, oh, well, this contains COP1, and COP1 is mostly useful, well, really pretty much only involved in ER to Golgi. Oh, there must be something there. Okay, so what's the purpose of protein codes? Pretty much, if you have to take home some message about protein codes, it's that it's involved in concentrating proteins in a certain area that's going to form the vesicle, and then forming the vesicle. So the actual conformation of these protein coats are curved, and so they help in forming the curvature of a vesicle, so that budding process. And so you can see here, you've got your different protein coats, and then you've got these receptors that are binding up the cargo. So those are the proteins that are going to enter the vesicles that are going to go off to the Golgi. And so they're going to concentrate all those proteins into the vesicle, and then it's going to bud out and then go to the Golgi. So the example I give you here is clathrin. So that's just one protein code, but the major concept is the same. So there's clathrin, there's the adaptor proteins, and then there's dynamin. Those are your major players. And so clathrin is going to be your coat that has that curved shape that helps with formation of your vesicle. Adaptin is going to bind to clathrin and to these receptor proteins that are going to bind the cargo. So you can see here, it's going to concentrate everything into the vesicle through interaction with those receptors. And so then they're all going to come together, they're going to form your vesicle, and then dynamin is going to tighten up. It's going to tighten up around the membrane and pinch it off into a vesicle, so then it can leave the ER. And once it leaves ER, then your coat proteins are going to release. So it's about concentrating the cargo into the vesicle and actually curvature of the membrane to form the vesicle. Questions?
Can you go over the last part with the ER? It leaves the ER?
Yep. So I was just asked to go over the last part. So it's going to leave. So okay. So you've got you're concentrating your proteins because they're interacting with those receptors, those cargo receptors, which are interacting with your adaptor proteins and clathrin. And so then you're going to get the Dynamin that's going to tighten around the ER membrane, and that's going to cause the pinching off of your vesicle. And then once it leaves the ER, the proteins dissociate. Yep, and so that's what you're seeing here. And so then you have this naked vesicle. So you can see the Dynamin is going to mediate that pinching off. Yes?
So how does the membrane deconnect? Like once the Dynamin pinches off, how come there's none?
Well, so that's just the nature of membrane, right? So the question is, how come once the Dynamin pinches off the vesicle, why isn't there a hole in the membrane? And so think about membrane and how it's phospholipids and it's this fluid bilayer. So it's not just like a sheet. It's these phospholipids that are all stacked together, and they don't want water there, right? Water's bad news. And so they're going to come together to exclude water. So it's kind of this automatic process. Does that make sense? Kind of like you can imagine if you pour some oil in a cup of water, right? If you have a big oil droplet and then you like poke it with a fork, it's going to want to come back together, right? Has anyone ever done that? Poured oil in water? Don't pour water in oil if the oil's really hot. You could own a stain stick if you. Half my clothes are like covered in oil splatter. I'm like, well, stain stick it. Sorry, no, but you know, like if you want to, so when next time you're cooking, if you want to think about how membranes work and oil and water. So this is showing the pinching off. And so these are just going to kind of come together, right? These are going to want to come together, and these are going to want to come together just by the biophysical properties of lipids. Okay, yep.
I just had a question to make sure I understand. Like, so this is all part of the endocytic pathway. So like, all these proteins are working there when the vesicle go back in.
This is the so remember, thank you for that question. So the question is this about the endocytic pathway? Well, so remember I showed you this, and I said endocytic and secretory pathway are kind of one big process, in and out. But it's all the same in that proteins move between compartments in vesicles. Coming in, going out, it doesn't matter.
Okay, so it's both.
Right. There are different codes that are functioning, but the general idea is the same.
Okay.
Yeah. So here's a figure just of your general mechanism of coated vesicle formation, and it's the same thing. So you've got your cargo sorting, cargo capture, maturation, and scission, which is the pinching off. So that's where your Dynamin's working. And I want you to notice that actually, if you're reading this, or you'll be reading it at home, you'll see GTPase. And I'm going to introduce shortly the concept. Ooh. What was that? Those are my daughters, but they're a lot older. They're not. That was like when they were 3. PowerPoint encountered a problem.